Hello everyone, good morning, good afternoon, good evening, and welcome to our today's webinar, Bayesian Approaches to Address Clinical Development Challenges for COVID-19 Vaccines, Drugs, and Devices. Let me introduce myself. My name is Natasha Müllerman. I'm Vice President in Strategic Consulting in Cytel, and it is my pleasure to moderate today's webinar. I'm very pleased to introduce our panelists, Rajat Mukherjee, Frank Harrell, Greg Campbell, and Bernard Fritzell. And now they will do a short introduction about themselves.
Hello, everybody. My name is Rajat Mukherjee. I'm a research fellow statistician in the strategic consulting at Cytel, and I work with innovative and flexible designs, and also with rare diseases and devices, and have been currently engaged in several designing of COVID trials, both for vaccines as well as interventions.
I'm Frank Harrell. I'm a professor of biostatistics at Vanderbilt University in Nashville, Tennessee, and I'm also quarter-time expert statistical advisor to the Office of Biostatistics at the Center for Drug Evaluation and Research at FDA. I'm currently working on designing a number of COVID-19 intervention studies with Bayesian design, and also very active in developing statistical methods for longitudinal ordinal outcomes with Bayesian models that are applicable to COVID-19 clinical trials.
Hi, I'm Greg Campbell, and I am an independent statistical consultant. Before that, I was for 20 years the director of the Division of Biostatistics in FDA's Center for Devices and Radiological Health. Before that, I was actually a tenured statistical scientist at NIH. And I have a number of clients now who have COVID kinds of applications for their devices, and I've worked with the drug as well, and some diagnostic companies as well. So it's a pleasure to be here.
Hello, good morning, good afternoon. Bernard Fritzell. I'm MD by training. I'm currently a consultant in vaccine development and vaccinology. Before that, I've been involved in vaccine development for more than 30 years at various companies, my last position in industry being Vice President for Scientific and Clinical Affairs at Pfizer Vaccines. I am a member of the WHO PDVAC committee and the WHO TB vaccine R&D group. And I've been involved more closely with the clinical trials in the COVID field, testing for the non-specific effect of BCG to prevent COVID infection. And it's a pleasure for me to be participant in this panel.
Okay, thank you for all the panelists for the introduction. So we start with our first discussion topic. Starting with today, we will cover vaccines, therapies, drugs, and devices. And starting with vaccine, looking at now when we are almost gone through the first wave of coronavirus pandemic, the immunity among the population is still low. And there is a lot of investment and efforts by governments, industries in finding the fastest possible way to develop a safe and effective vaccine. Usually vaccines development is lengthy and expensive process. How is it compares to the development pathway for vaccine under the new pandemic? What are the possible pathways to shorten and to speed up the process?
Okay, the development of the SARS-CoV-2 vaccine is facing several challenges, which impacts its complexity and its time duration. There is no established immune correlate of protection. Neutralizing antibodies are expected to be key player in the immunity and the Th1 response to strengthen this protective immune response. There is no validated predictive animal model. And last, there is no controlled human infection model that would allow rapid assessment of the vaccine. So overall, there is no way to demonstrate vaccine efficacy outside of a classical efficacy trial using clinical endpoints related to the COVID-19 disease. As was just mentioned, the classical clinical pathway of vaccine takes about 5 to 10 years, with a substantial fraction of this time duration being related to the conduct of the phase 3 efficacy trial. And for this phase 3 efficacy trial, a critical factor for feasibility and success is the attack rate, the hazard rate. And it needs to achieve a certain level. So study population are being selected for being at high risk of exposure. This is a case of healthcare workers. However, there is uncertainty and unpredictability of virus circulation and transmission. And in addition, the impact of increasingly implemented preventive measures need to be taken into account because it's likely to reduce the attack rate. And this is very important when you think about the feasibility of a study, because whether attack rate is 2 or 20%, you will multiply by 10 the sample size required to demonstrate the efficacy of the vaccine. So the purpose of the WHO Solidarity Vaccine Trial is to offer a large multi-site international platform study for phase 2B and 3. At each site, there should be high enrollment capacity. There should be structures and procedures to ensure good quality follow-up for safety and efficacy assessment, good retention of the study participants to minimize the loss to follow-up, and last, more important, the capacity to diagnose SARS-CoV-2 infection. And the purpose is to allow simultaneous evaluation of several eligible vaccine candidates through an individual randomized controlled trial using as a primary endpoint virologically confirmed infection. It should allow the concurrent risk-benefit assessment of each selected vaccine candidate within the time frame of 3 to 6 months, with the objective to drop very early on those candidates, vaccine candidates that are not fulfilling efficacy or safety preset criteria, and to focus subsequently on those promising candidates which would have shown in interim analysis a relative benefit over the placebo or and over the other candidates. So for that, adaptive design with decision driven by interim analysis facts should allow the sample size re-estimation depending on the attack rate, because this is a major uncertainty when you design the vaccine efficacy trial, and also to consider randomization reallocation between the active and the test groups, so that as soon as efficacy trends could emerge. And I think that now Rajat will tell us about the most suitable statistical approach to address these clinical questions.
Sure, thanks, Bernard. So Bernard, you spoke about adaptive designs there, and yes, it's true that adaptive designs are particularly well suited when there are uncertainties around design parameters. For example, what is the effect size? What is the statistics for your control arm? Whether it's a vaccine trial or an intervention trial. What is the variability in the endpoints that we are interested in? So all these uncertainties, in order to mitigate risk and even to maximize the chance of success for a particular trial, adaptive designs have been used. But we are in a situation where, we are talking about the extreme, the lack of any prior data or knowledge about these design parameters that would influence things like sample size or the duration of the study. And I think in these cases, Bayesian designs offer a lot of flexibility. Flexibility not just in terms of sample size. We all know that in frequentist regimen for adaptive designs, one has to pre-specify the sample size. In Bayesian, there's more flexibility in sort of designing a study with a flexible sample size. You might decide, from during interim analysis, whether you want to stop enrollment or you want to proceed with the enrollment. But also not just the flexibility in sample size, but also the flexibility to have frequent interim looks, especially if you are looking at a trial where the expected recruitment is really fast. It will still allow you to take some frequent looks at the beginning of the study in order to minimize overruns, and also to look at both safety and efficacy signals to then sort of predict what would be a suitable sample size to come up with the evidence that will convince the community, the scientific community, the regulators that a particular vaccine or an intervention is working. It's obviously, having said that, like any other adaptive design, either in the frequentist genre or in the Bayesian framework, one has to carefully evaluate the operating characteristics. And I think, we should not talk about type 1 errors in the Bayesian framework. We should probably, talk about false positive or true positive rates with respect to, obviously, the data generating process. Having said that, I think I'll pass on the floor to Greg to talk a little bit more about the utility of Bayesian designs in a situation like the COVID pandemic.
Thank you very much, Rajat. Before I get into that, let me broaden things a bit from vaccines and talk about COVID trials in general. And a good source of information about all of the clinical trials that are ongoing is the registry that's put together by Cytel and the Gates Foundation called Clinical Trial Tracker. It now has more than 1,100 registered trials. About 27 or more of them are vaccine trials. There are more than 200 in the US, and in Europe there are even more. And most of these are small trials. They're not very large. Most of them are under 100 people. But there is another source of information. Clinicaltrials.gov also has the ability to search trials, and what it shows is, yes, indeed, there are more than 1,000 COVID trials. About 121 of them are device trials, of which 11 are diagnostic devices. The substantial number are, however, drug trials, but some of them are preventative trials as opposed to therapeutic trials. Interestingly enough, in the search of clinicaltrials.gov, there are only 2 Bayesian trials. And I look at that with a bit of skepticism, because throughout, as we've been thinking about Bayesian trials, I've gone to that source and looked at trials that I knew were Bayesian, but Bayesian is not in the description of the trial. So my guess is there are a lot more trials that are Bayesian. As Rajat said, one of the big advantages is the ability to adapt, and Bayesian trials must be extremely adaptive. But one of the things I think that makes it so attractive, particularly in an effort like the WHO effort for the solidarity trials for treatment, and then, as Bernard mentioned, the solidarity trial for vaccines, is the ability to set up what's called a master protocol. This is, and some people call this a core protocol, but it's an overarching design to evaluate multiple treatments or multiple therapies. It improves the efficiency and establishes uniformity throughout using standardized procedures in the development and evaluation. And those can be things like, you know, how do you recruit patients? What are the inclusion and exclusion criteria? Providing standards for data management and for analysis. And so there are a number of master protocol trials that have been done for COVID. The early ones for Ebola, the second one that was done in the Democratic Republic of Congo was in fact such a trial. It was a master protocol trial. And one of the advantages is that, particularly with regard to infectious diseases, is what do you do if when you're trying to recruit patients, the infectious disease stops, which is to say, it's a good thing, but you're not at the end of the trial. What do you do in those cases? And the master protocol can allow for the fact that you could quit and then resume, perhaps in a different location, or perhaps when COVID resurrects itself in the same location. So that's a big advantage. The other thing that I wanted to say is most of the master protocols involve what are called platform trials, and that's very much useful in COVID kinds of situations. Here the idea is you have multiple therapies that you want to study at the same time for, in this case, one indication. Platform trials, that's called an umbrella trial. You can also have what are called basket trials, but that's where you have a single therapy in multiple diseases. But one of the advantages of platform trials is they're usually randomized, and there's usually a common control group that everybody compares to, that the different treatments don't compare to each other. That's not the intent, but they compare to the control, the common control. And this idea of master protocol and platform trials is really originated in oncology, oncological drugs, and has been embraced by FDA. Janet Woodcock and Lissa Lavange in 2017 wrote an article about master protocols in the New England Journal of Medicine. So there have been some good examples historically of Bayesian trials that are platform trials. I-SPY 2 is one of the most famous, with Laura Esserman and Don Berry. And then there's a glioblastoma multiforme Agile trial, which is also very famous. So those are some examples of early efforts to use Bayesian statistics in platform trials, but it's really quite helpful because it allows you flexibility to, for example, drop an arm that's not working, potentially to introduce a new intervention or a new vaccine if one becomes invented after the trial begins. And so that kind of flexibility, particularly in a situation of SARS-CoV-2 or COVID, is extremely important. And so that's where I kind of like to, I think we'll have much more discussion about the use of Bayesian statistics for these kinds of trials. And I look forward to the discussion.
Okay. Now, thinking of if we expand the discussion from vaccines to also the treatment trial, maybe we could discuss the difficulties related to the endpoint selection and how we can maximize clinically, like how we can use all clinically available information. One of the frequently used endpoint is the WHO severity scale, which has which allows to demonstrate really the clinical value of where the patient is in terms of severity. But if we select predefined time point, we might miss information in between these time points. And we will not really necessarily capture probably the most sensitive information. If we're thinking of the time to change, the other question is also how relevant is the shift, and what is the relevant, clinically relevant shift? Is it 2 days, is it 3 days? Thinking of more severe patients who will go in respiratory failure and who might require CU admission, invasive mechanical ventilation, we probably also would need to consider the whole course of the disease, because some patients, we might measure time to recovery, but some patients first deteriorate and then recover. So how we can capture, what would be the statistical method which would allow us in different situations leverage the most of the clinical information we could accumulate?
Maybe I can make a comment there, because in the vaccine field, the development of a vaccine will be to prevent the infection and the disease. Of course, and this will be the primary, likely the primary endpoint in this master protocol that Greg was alluding to. If you want to assess the impact of the vaccine on the severe cases, you will also have to include that in your sample size calculation, because if the sample size calculation will be based on the primary endpoints, which will be anticipate biologically confirmed COVID infection or disease, whatever it is. And only a small proportion of that, maybe depending on the age, 2 to 5%, will come into a resuscitation unit or require intensive care. And so, of course, in the trial, you should collect and have this information, but maybe not. I would be a little bit cautious about the information and the learning we may take from that, because there will be very minimal, not too few cases.
Yeah, I think that certainly depends on what is the trial objective on population. If we're talking about prevention, or we're talking about patients who are already hospitalized and already in respiratory failure, yes, then there will be different needs and the different primary endpoint.
Yeah, here I think my understanding for the vaccine development, it is a preventive approach.
So let me jump in here and just talk about endpoints in general. I think in the first Ebola trial in West Africa called PREVAIL-2, the endpoint was 28-day survival. And the reason for that, for Ebola, is that what's called the case fatality rate, the proportion of people who die who've been diagnosed as positive for Ebola is more than 60%. And so the endpoint there is mortality, 28-day mortality. And that trial was a Bayesian trial. That was a trial sponsored by NIAID with Laurie Dodd and Mike Prosham. And they termed it barely Bayesian because they used a non-informative prior. And the trial didn't work because the Ebola virus died out before they could recruit everybody. The other interesting thing was they followed that up with the PALM trial, which was not a Bayesian trial in the Congo, and they used 28-day mortality as well. But they had 10-day mortality, and what they found was the 10-day mortality predicted the 28-day mortality. As a Bayesian, you could build that into the model. And so you could figure out how predictive 10 days were to predict mortality at 28 days. And I don't know if they did that formally or not, but my guess is that that was part of the thinking, to be sure. And you can do that if you're a frequentist, but as a Bayesian, it's just really easy because you end up estimating the parameters to predict the 28-day mortality from the 10-day mortality. The interesting other thing about endpoints is that the NIAID trial ACTT, which is an adaptive trial but not a randomized trial, they changed the endpoint. And that's sort of an interesting thing. If the data are blinded, whether you're a Bayesian or not, you can change the endpoint. And they, in fact, did that. And so instead of, I think originally they had planned on mortality, but they changed it to time to hospital release. And were able to then show that remdesivir reduced the time from 15 days to 11 days, but they didn't meet the mortality endpoint, although there was a hint that it was better for the remdesivir arm than the other arm. So the whole thing about endpoints depends on how the virus operates. And in this case, for COVID, you have a different situation than Ebola. So it's not so clear that mortality alone is the only thing that you need to think of. And that's a reason to think about the 7-point ordinal scale that WHO was recommending.
I'm glad that Greg brought up several general issues related to endpoints. Those are wonderful issues. And one issue that a lot of non-statisticians don't tend to understand is that outside of the Ebola situation, where your event rate is not very high in the usual clinical trial, time to event analysis doesn't have much more power than just a binary endpoint analysis. And a binary endpoint analysis has the minimum power of any analysis you can name. And so there's still this wish among non-statisticians to do responder analysis. And when your time to event is really time to something good happening, such as time to hospital discharge, how you handle death gets really complicated. And I forgot how they handled it in that particular case, but another recently published trial at New England Journal of Medicine handled it incorrectly, just censoring on death, and you can't do that. So I'm always searching for what is the statisticians review. They view responder analysis as like one of the #1 sins against data. What would be the medical equivalent? I think the medical equivalent to responder analysis would be using antibacterial on a virus, or it would be giving a 2-year-old the same dose as a 40-year-old. It's a responder analysis ignores so much information, and it has just every statistical problem under the sun. So I think we know in general in clinical trials that longitudinal data has our richest source of information, and using that to the fullest to get maximum power, as well as to answer, I think it was Rajat mentioned, you're wanting to find out when is the treatment effective, and not assume that if you picked one time like 14 days, that's really the time to pick. So if you have longitudinal data, you have much more power, and you can answer all the questions because you can estimate like time and state, you can estimate transition probabilities, you can estimate all sorts of things. And you don't have to be right about choosing the time. And the other thing about longitudinal data is if you measure it frequently enough, your problems with missing data go significantly down. So the more frequently you measure, say, an ordinal outcome, let's say you're able to get it daily, if somebody drops out and they're not, and they're censored, you'll have a recent measurement. So you're more likely to assume missing at random and be okay with it if you measure frequently and not just wait to one measurement every week. So I think there's a lot of issues related to endpoints that have come up in the discussions of COVID-19 clinical trials. It's been a fascinating discussion, and I think the people that are wanting to do responder analysis need to be doing some power simulations, and they'll never want that again.
Greg, just to add to your comment on longitudinal analysis and collecting the data longitudinally and frequently, I think another main advantage is in a pandemic situation, I think it's really important to also be able to evaluate the impact on public health. resources. So if you collect that data, let's say you are looking at a WHO scale and you are looking at a bi-weekly or a weekly data, then you should be able to evaluate what is the early impact. So you could still look at a 15-day outcome as your primary endpoint, but if you collect that longitudinal data, you should be able to evaluate the early impact of a particular intervention, for example, for severe cases.
We have a question from the audience on the endpoint. What about viral load reduction as a primary endpoint? What does the panel think about it?
There have been some thoughts on using shedding as an indirect way to assess the indirect effect of the vaccine. But I think that I'm not so sure that today the way to assess the viral load in a study subject and to in a validated manner would allow to in a vaccine study to bring information. You would have to sample the study subject nearly every day or every 2 days to see how it is evolving, because you must see what shedding is present. And so it's quite difficult to document that, at least in the pre-licensure studies.
And I guess also based on what we just heard, the discussion between Frank and Rajat, right, on the maximizing the information. If you look at the viral shedding or viral load, it almost would be then binary, yes or no, because it will be quite difficult probably to determine the degree of that, right? So it's almost becoming again the binary endpoint, and it's still not directly clinical outcome.
But as Greg mentioned, it could be definitely used, for example, predictive power calculations at interim analysis, if that information was available. And I think it will definitely means basically what I'm trying to say is Bayesian designs also at interim analysis, you take in the totality of the evidence that is available at that time. So if the viral load kind of information was available, then I think we would, as statisticians, definitely be happier to have more rather than less.
We have another question from the audience. What could be the endpoint for immunomodulatory repurposed drug? So here we're evidently talking about treatment, right? Treating COVID rather than preventing. That's what I would assume from the question. Yeah. So we're coming more to the treatment endpoints, which would be the most informative.
In a preventive manner, I don't see. I'm used to think in terms of prevention. So certainly that once the exact or refined profile of the immunological features associated with the severe forms is well defined, then you could consider to better defined endpoints, but I'm not.
Because we see in the literature and the discussion, if you're thinking of immunomodulating intervention, I guess the question first of all is what is the right moment? If patients are becoming already critically ill, is it too late to intervene? Should it be done when they are at, let's say, hospitalized but not yet in critical illness, so moderate to severe but not yet critically ill? Because depending on what population would be the target, critically ill, not critically ill, then the endpoint would be quite different. But still, if we really want to show clinical benefit, they have to reflect. They have to be clinical outcomes.
maybe the progression would be possible when there are identified correlate of risk to develop severe disease. So you could identify in the patient population those presenting this correlate of risk, and then consider them as a target population for being for the assessment of such immunomodulator treatment. But I'm not aware that validated biomarkers of that risk have been identified.
Yeah, I think that's, at the moment there is no really markers, at least, which would be published to be used. And then I think coming to the question of the endpoint, it depends on what the drug is supposed to do. If we take moderate patients, for example, and we want to prevent respiratory failure or prevent deterioration, then we should be more looking at preventing the degree of the severity, advancing into the severity of the disease. But if we're talking the most critically ill patients, if we're in a critical care setting, or certainly mortality would be important, even for it might not be the primary endpoint we would expect drug to act, but at least we would need to show that mortality is not impacted in a negative way. And then we could look at, for example, time of requirements for invasive ventilations, days on ventilator, which is frequently used endpoint in critical care, and eventually overall length of stay and recovery. But it really depends on what is the main hypothesis behind the drug's efficacy. Now, discussing the Bayesian design, another question we got from the audience, I think to some extent you would cover it, but it probably would be helpful to synthesize it. Bayesian design is especially known to be very useful when there is a prior information, right? And here we're talking about condition without prior information. So you already mentioned a lot of other benefits from Bayesian, that maybe we could just summarize it of not having prior information where you would traditionally think of Bayesian, how would still be useful compared to frequentist adaptive approach.
Right. So in 2010, when I was at FDA with me as a leader, released a guidance document on the use of Bayesian statistics in medical device clinical trials. And that document outlines several things. One is to make decisions based on the posterior distribution instead of relying on hypothesis testing, p-values, and things of that nature. But one of the others was sort of thinking about it in two separate ways. One is in situations where you have prior information and you want to use it, and this could be subjective prior information or it could be more objective. And in those situations, you can use statistical modeling like hierarchical Bayes modeling or power priors to incorporate quantitative information from other randomized trials. Now, in the COVID situation, I don't think we really have that kind of prior information. And so it's really the second kind, which is the Bayesian adaptive designs outlined in that guidance document that come to the fore, I think, in this vaccine emergency. And so the idea there is to use a prior that's not informative, that you're not relying on any prior information. You could do a Bayesian adaptive design with prior information, but in most cases, people do not do that. And what they do is they allow the information that you accumulate during the trial to be able to leverage that information to make decisions earlier. And part of that is the ability to model predictively what's happening. And so not only do you have the posterior distribution to help you, but you can predict at any time based on information you collect longitudinally, as Frank points out. You can use that information to predict what patients who aren't at the final stage, who maybe even haven't entered the trial, but are perhaps at some intermediate stage, what their final endpoint might look like. And that turns out to be very, very useful. And that, I think, is the real strength of a Bayesian adaptive trial, even though you don't have any prior information. And it's true that if you're a frequentist, you can do that, but I think it's just much harder, it's much clunkier to be able to try and do that. So that would be my contribution to this part of the discussion.
Just to add to that, Greg, I think in recent times, when we do adaptive designs, for example, decisions that are based on conditional power calculations at interim analysis, we augment the conditional power frequentist calculation with Bayesian calculations of predictive power in order to take a much more, I would say, a decision with confidence.
And just to add a few more advantages of Bayes, one that most people don't think about is your calculations are exact, whereas in the frequentist world, when you're not using normal regression, most of our calculations are fairly approximate. And then we've talked a lot about flexibility in learning, so the ability to have unlimited looks at the data with no penalty at all is what makes Bayes have the ability to learn more rapidly, jettison unpromising treatments, and extend studies when you have an equivocal result. Handling multiple outcomes is a big plus with Bayes because you can calculate joint probabilities connecting the outcomes, like what's the probability that you lower mortality by any amount, or you lower the chance of needing a ventilator by a clinically significant amount. And speaking of clinically significant, the ability to bring clinical significance into the probability calculation is kind of unique to Bayes. And so there are a lot of advantages, and I think the ability to avoid multiplicity problems is one of the single biggest one. And multiplicity comes from data multiplicity, and the more data you acquire and the more looks at data you make, you're going to have a higher chance of finding extremes in the data. And the Bayesians say that's completely irrelevant because we're not interested in data probabilities, we're interested in efficacy probabilities.
Right, because yeah.
Natasha, can I just add one more thing about use of non-informative or informative priors? So there could be situations. So I think ideally one would think of a seamless design. So seamless learn to confirm Bayesian design in a pandemic situation, whether it's vaccine or intervention. But there could be situations, for example, you could break up a clinical trial into 2 phases or even 3 phases. In the earlier phases, you look for safety signals, right? And then you want to sort of zoom in into what is your primary endpoint. And you may not be clear about what your primary endpoint is until you see some data. At that point, once you have decided, okay, this is the primary endpoint, now we go to the confirmation stage. You could still start that confirmatory stage with an informative prior, and that will definitely help reduce the sample size as well as speed up the study.
So now opening the question of multiplicity and the seamless design, but very frequent question we are getting is how, if we're talking about phase 3 pivotal confirmatory trial, would regulatory agency accept Bayesian design, or how we would make Bayesian design acceptable for regulatory agencies, speaking especially about FDA and EMA?
So I guess this is where I chime in, having spent 20 years at FDA. In the device part of FDA, we've had great success with Bayesian trials, both with prior information and without prior information. The prior information discussion, though, becomes much more complicated, and we usually need to bring in our experts, clinicians, and engineers in the case of devices. For the question, though, I think it's helpful to think about 2 different kinds of trials, and one is the exploratory trial where you're trying to learn things, and in a situation like COVID, that's really important. I mean, and a lot of the platform trials are trying to figure out, which of a number of interventions should you go forward with to the next phase. And so I-SPY 2, for example, that was the intent there. The intent was you would look at different therapies for breast cancer, and as the therapy matured and showed that it showed promise, it would be graduated to the next phase. When you talk about confirmatory trials instead of these exploratory trials, I think you have to be a little more careful, and regulators, I think, are pretty careful about making sure that they don't make a decision that they're going to end up regretting. And one decision would be that you falsely approve something that's really not effective or really is not safe. And the reason for that is once that is done, it's very hard to put it back in the box once it has escaped. And so for a lot of reasons, people in regulatory settings, in confirmatory trials, are very careful. And so in the Bayesian guidance document that I mentioned earlier, we encouraged companies to simulate operating characteristics of trials, to learn by computer simulation under various scenarios what would happen in terms of sample size, what would happen in terms of false assertions, type 1 error, what would happen in terms of power. And you learn a lot, I think, by doing that as a company, and FDA learns a lot as well. And what we learned at FDA was that often companies would not pick scenarios that were they didn't have a broad expanse of scenarios, and often we would ask companies to look at other simulations. What would happen if the device really wasn't quite as good as the salesman for the companies might want to make it seem? And so that's where I think regulators have to be a little more careful and think about things like multiplicity in ways that a true dyed-in-the-wool Bayesian might not want to think about. But I think from a regulatory standpoint, it does make sense to ask questions about, how often am I going to falsely assert that this drug works when it doesn't work? And so I think that's the challenge.
I think this is where the controversy really starts. And a great philosopher said, in time, all things shall become unclear. And I think the more you think about type 1 error, the more unclear it gets. The shortest analogy that I have for what type 1 error represents is talking. And so what's the probability somebody's going to talk? And that has almost nothing to do with the chance that they're telling the truth. And so if you judge somebody's veracity by how often they spoke, you would be a very poor judge of veracity, because you would want to look at the probability that they speak the truth. And so we really have a psychology of accommodation. And with all respect to Greg, and he knows, he and I have had this discussion many times, but it's the psychology of accommodation, because the question you want to answer was not answerable when Neyman and Pearson and Fisher were inventing all these techniques that we have, and we didn't have computers. And so we needed probabilities that could be calculated with pencil and paper. And the probabilities that are calculable are ones that use a simplifying assumption, which is that the treatment has no effect. And that was never the probability we wanted, but it's become something we've accommodated to. And I've heard famous statisticians say something that Greg was careful not to say just now, which is that the type 1 probability is the probability of making a mistake and concluding that a drug works. And type 1 error could not be more different than that statement. And so I think what's happened is that people that cling to showing frequentist operating characteristics, when what they should be doing is showing Bayesian operating characteristics, they're actually clinging to something that was never true for them. And I've talked to several leaders at FDA who have grossly misunderstood this, especially non-statisticians, who have said type 1 errors are probably of making a mistake in saying that a drug works. It's not even related to that. So it's a probability of talking. And I think the sooner people realize that type 1 error never bought for them what they had purchased, it never solved the problem of telling you about false positive or false negative rate, which I think Rajat might have mentioned at the outset today. I think we'll start to make progress. So the Bayesian says you need to show that your posterior probability is well calibrated, and the only way it's not well calibrated is if there's a person in position to judge your work who has a prior that's their own prior that they never told you about, and their prior would override everything that was done in planning the study. So that to them, the posterior will be poorly calibrated. But other than that, you don't want to look at type 1 error because it's not the decision-making error in the least.
So for the regulator, if I get your answers together, right, it looks like the simulations are really important, right? If we plan Bayesian trial and if we want to go to engage with FDA and EMA, it looks like companies would need to think and invest more upfront to run more thorough simulations, but they would get then the benefit of all the flexibility and adaptation from the Bayesian when they're going to be running the trial. So a little bit more investment upfront, but benefits coming through.
That's true, and the more you talk to FDA, the better things go. And the big picture is there's a lot of modern, forward-thinking people at FDA. I've been amazed, and I work mainly with the statisticians, incredible statisticians, and they're really know modern methods. They write a lot of great papers, and I think the pharmaceutical companies project onto FDA a conservatism that FDA doesn't really possess, although it is dependent on who you get assigned as your reviewer at FDA. I found that the pharmaceutical companies, except for 3 notable exceptions, are much more conservative about this than FDA is.
I would like to thank our panelists for the discussion, and thank you to the audience for attending and for submitting your questions. So thank you very much, and I think now we can close this session. Thank you very much again, panelists and the audience.
Thank you.
Thanks for having us.